Our second presentation of the morning is for the Redfield Lifetime Achievement Award. And I'm really honored to introduce to you the recipients of this award, Drs. Evelyn and Barry Sherr, who are professors emeritus at Oregon State University. So we have kind of a clean sweep today by one institution. So this award honors major long-term achievements in the aquatic sciences, including research, education, and service to the community and to society. And Evelyn and Barry are receiving the Redfield Award for their joint lifetime research on food web interactions, protists, and the factors controlling the activity, growth, and survival of aquatic microorganisms. And the Sherrs are really a unique and dynamic team. They've been working together for almost 40 years and have in many ways served to transform the field of aquatic microbial ecology. And while they are each, in their own right, outstanding researchers, their longtime collaboration has enabled them to achieve a higher level of excellence. This is something I found on one of your web pages. They say they do mom and pop science because they work together in all aspects of their job, including mentoring grad students, going to sea to collect data, analyzing data, and writing papers together. So they're undeniably an inseparable duo. And their research into food web interactions of aquatic microbes has been instrumental in forming our present-day understanding of the microbial loop and the role of protists in the aquatic food web. They've investigated microbial food web interactions all around the world, including Lake Kinneret in Israel, the Mediterranean Sea, coastal waters off the southeast and Pacific Northwest areas of the U.S., and also the Arctic Ocean and Bering Sea. And of particular note is their development and use of innovative analytical techniques, including CTC staining, flow cytometry, and cell signaling to answer their research questions. And beyond their impressive research careers, Evelyn and Barry have been selfless mentors and continue to be selfless mentors to graduate students and early career scientists. They've served on several editorial boards and are advocates for research in broad areas such as global warming and evolution. So please join me in congratulating Evelyn and Barry Sherr for receiving the 2016 AC Redfield Lifetime Achievement Award.
Okay. Can you hear me? It's okay? Good. Okay, so this is a fairly low-key talk. I don't know how to summarize over almost 40 years of research, so it'll be sort of fun. Anyway, the talk is based on our favorite group of organisms, planktonic macrotrophic protists. And I must mention that we have put our talk, plus amongst other slides, as a Google photo album online, and you can get to it from my Facebook page, or if you want to just email us, I can send you the link. So you don't have to take notes or anything, and you can study all these slides at leisure if you want to later on. Now, we were totally surprised by this award. We're very appreciative for all the hard work of the nominating committee, our student Krista Langnecker, who organized this. Complete surprise to us. We definitely appreciate the recognition, not only for our work, but also the work for our colleagues who have worked with macrotrophic protists along with us over the past few decades. And we owe a ton of gratitude, appreciation to all of our mentors, students, postdocs, and colleagues we've worked with over the years. Now, this photo, it has a history. This was an image I used in one of the very first talks I ever gave at a conference, so I had to work it into this one too. If you go online, you can learn what exactly it is. I won't tell you now. Now, One of the first suggestions we had about the role of phagotropic protists in aquatic food webs, mainly marine pelagic food webs, but also relates to freshwater lake food webs, was the idea that phagotropic protists repackage smaller cells into larger cells, the flagellates and ciliates. We didn't understand about heterotrophic dinoflagellates at that time, and then served as food for larger organisms, the metazoa plankton. So we call this fixed carbon repackaging and recovery pathways, spaghetti diagram here. And this was actually a comment in Limnology and Oceanography in 1998. We were very pleased to see last year, 2015, Alexandra Wharton and colleagues produced a much more elegant diagram of sort of suggesting the same thing as a review in Science. It's a really nice paper. Anyway, you can see that the heterotrophic protists, again, are right in the middle of things, and they express the same concept that they're repackaging smaller cells toward the mesozooplankton and eventually onto higher trophic levels. Unlike in lakes, in marine systems, most metazooplankton can't really effectively graze on cells smaller than about 5 microns. So the protists are grazing on picophytoplankton and bacteria and then serving as food for the higher organisms. Now, just a bit of background if you don't know a lot about heterotrophic protists or marine pelagic food webs. I want to give a bit of background about why they are important. And the first thing is that these organisms mainly are heterotrophic, they're colorless or non-pigmented, and they occur in 2 size categories. This is a famous diagram of Finchel et al. In Illinois in 1978, and plankton ecologists have used these order of magnitude size categories going from viruses all the way up to whales to categorize different groups of organisms. You can see that the bacteria are in the 0.2 to 2 micron. Phytoplankton have a very large size range from almost bacterial size up to very large cells and chains. And the protozooplankton, back then they called protozooplankton, we've since switched to protist because protozoa means first animal life, and these guys are not related to animals whatsoever. So now we just call them protists. They overlap the phytoplankton. They're mainly in the nano and micro size classes, 2 to 20 microns and 20 to 100 microns, but they do slop over into the picoplankton, and some are larger than the 200 micron size classes. And then the metazooplankton higher trophic levels are in the higher order size classes. The nano size is mainly phagotrophic protists, flagellates. Many of them are completely heterotrophic. For instance, the choanoflagellates, which are completely heterotrophic. None of them have any kind of chlorophyll or photosynthesize. And then a cafeteria species, which is a heterotrophic chrysophyte, is related to autotrophic cells, although it itself does not have any chloroplasts. But there are now, we now know, many phytoflagellates which are ACDC. They are called mixotrophs because while they have chloroplasts and photosynthesize, they also ingest other cells, mainly bacteria. And the other size class is in the micro size class, 20 to 200 microns. We usually call this size class microzooplankton, although they're not related to crustacean zooplankton at all. Many species of ciliates and heterotrophic dinoflagellates. They prey on phytoplankton, including diatoms. And they're major herbivores in the sea, really important herbivores in the sea. As an example, heterotrophic dinoflagellates include thecate ones, like this protoperidinium, which is feeding extracellularly by a feeding veil that comes out of the cell and spreads over this diatom chain and is digested extracellularly. But there are also a lot of heterotrophic dinoflagellates which are non-thecate and which can engulf prey cells. And then ciliates, there are a lot of different kinds of ciliates in the plankton, including non-testate ones, but also testate tentinids, which are an interesting group in themselves. Why they are important? They're important for three reasons. One, they eat. They eat a lot of different things of all size classes. The little nanoplankton protists eat bacteria. They're very important in eating bacteria, but not only heterotrophic bacteria, but also autotrophic bacteria, the coccoid cyanobacteria and prochlorophytes. We know that some work has been done that shows they can even ingest smaller things like viruses and high molecular weight organic matter. And they eat eukaryotic phytoplankton. All size classes of protists eat eukaryotic size phytoplankton, all size classes of them. And I'll show you some examples, pico to nano to microplankton size. And with all the eating, they poop out a lot. They excrete. They're very important in remineralization in aquatic food webs. They excrete NMP compounds, inorganic ammonium phosphate, organic compounds, principally urea, amino acids, and organic phosphates. But they also do have some uningested particulate, very small microfeces that they also poop out. And because they have a fairly high biomass, they are eaten. They're an important food resource for metazoans. And here's an example from our Arctic Ocean work. This is in collaboration with Karen Ashton and Bob Campbell. We got a fecal pellet. They did grazing studies. And here's a fecal pellet from a small copepod, Matridia longa. There's the fecal pellet, and right there is the empty theca of a Protodidium dinoflagellate. Now, it turns out, serendipitously, that we have summarized our career about phycotrophic protists, work of ourselves and others, by a volume that's coming out, edited by Pat Liebert and Ton Canna. And they had the idea to do a volume based or focused on dual career couples. So our chapter in the book, not surprisingly, is on phagotrophic protists and microbial food webs. Now, they asked us to write a little blurb about our career as a couple. And our blurb starts out, we bonded over sewage sludge. This is true. Our first project together involved a study to look at the fate of nitrogen in sewage sludge spread over a Spartina salt marsh. Now, here's a career lesson for you. Don't overlook these very strange sources of funding. We got 2 PhD theses out of this project. And it was a sort of a strange project, but we got some good work out of it. We carried out the project at the University of Georgia Marine Institute on Sapelo Island, which is a barrier island on the coast of Georgia. You can only reach by boat. And we should also say that neither one of us started out working on protists. My PhD thesis research was looking at outwelling of nutrients from coastal salt marsh estuaries onto the continental shelf. And when I went to Sapelo, I also did some work on using stable carbon isotopes to evaluate food web interactions in the salt marsh estuary. Barry's thesis, which he did funded on that sludge project, was on denitrification in the salt marsh, looking at the fate of the sludge nitrogen with respect to denitrification. And he was one of the first ones to develop the acetylene inhibition method of looking at flux of denitrification by cutting off the last step. So you could get accumulation of nitrous oxide and measure that sensitively by gas chromatography. So here's me. This is the kind of salt marsh we did our work on. This is me actually, so I did sampling in the salt marsh. It's not the actual plot. So how did we become interested in protists? And this was a matter of luck and serendipity, another career lesson. We went over to Israel on a binational science grant to work with Tom Berman at his lab on the Lake Kinneret, which is also called the Sea of Galilee. And we were supposed to do work on nitrogen cycle process in the lake. Now, the particular work he wanted us to do didn't pan out. We couldn't actually get what he wanted us to do to work out in the lab. But we also found out that after the end of the annual dinoflagellate bloom in the lake, there was this bloom of colorless microflagellates, and the question was, what were they doing? So Tom said, okay, let's work on the colorless microflagellates, and that's how we got started working on phagotrophic protists. So a career lesson, if something doesn't work out and another opportunity comes up, go for it. And we did get a chance to tour around Israel. Here's one of our trips down south. We camped out on the shore of the beach at the Gulf of Eilat, which is the northern tip of the Red Sea, and there was a camel roaming around our campsite, and Barry's making friends with it. I won't tell you about the time we accidentally drove into Gaza City and the Gaza Strip. Okay, so then after we finished working with Tom on Lake Kinneret, we went back to ASLO and got into protist research full time. I should mention that there had been another study on protists by Robert Johannes back in the 60s, where he looked at the influence of bacteriolous flagellates on phosphorus regeneration in this same estuarine system. And I should say that Bob Johannes was Barry's first thesis advisor, and Johannes was working with Larry Pomeroy, who was Barry's second and final thesis advisor. At that time, there was very little known about phagotrophic protists. That was a cutting-edge research field, and there were very few methods. So the first challenge we had when we went back to ASLO was to develop methods. Now, epifrescence microscopy was just beginning to be used, for instance, to enumerate bacteria. So this was also a cutting-edge technique. So our first challenge was to adapt epifrescence microscopy to study of these protists, not only their abundance, seeing them in situ, but also rate processes. So we developed a method to look at the abundance by using DAPI as the dye, which turned out to be the best dye to use. We developed a method for fixing the flagellates so they wouldn't, when they had ingested something, some fixtures made them barf out the contents of their food vacuoles, which wasn't very good for doing the rate process studies. And then we developed the FLB method to look at the rate of ingestion. Tom Berman actually suggested the dye we used for the FLB method. So we did a lot of that work. Here's two examples from our studies with the FLB method. It's a small heterotrophic flagellate, and most of the protists ingesting the bacteria, same bacteria, were little flagellates. The food vacuoles, so here's DAPI, and then we changed the filter sets to see the green fluorescing FLB, and the food vacuoles are full of FLB. So we were able to get cell-specific grazing rates, extrapolate to population grazing rates, and then get an idea of impact, total flagellate impact grazing rates on the bacteria. We also saw ciliates, very small ciliates in the system, eating the FLB, which was a surprise because at that time ciliates were only supposed to eat phytoplankton. And we had little to do over that, but it turned out there were ciliates and they were eating bacteria. Here's one pelagic ciliate with about 40 FLB ingested in its food vacuole. And because we found the ciliates eating FLB, other people want to know were the ciliates eating FLB in their system. We got a NATO travel grant to go over to the French Mediterranean to work with Ferdinand Razadagan at his lab to look to see if his ciliates also were ingesting FLB, and they were. And we also got interested along the line about see if we can adapt this same process to look at cell-specific ingestion of phytoplankton, so protists eating phytoplankton. We got into it by the FLB method, so we made FLA, fluorescent labeled algae, and sure enough we could then document protists ingesting FLA. Now this is an example of mixotrophy. There's a typical, A typical pelagic ciliate, Strombidium ciliate, seen in DAPI. You can see the whole cell, and this blue mass is the nucleus, which is intensely stained with DAPI, which is a DNA stain. Switching filter sets to the blue light filter set, you can see this ingested fluorescently labeled algae. We had two different cultures, one 3 micron, 1.2 micron size, and these are two different cells and two different experiments, and they both show that they ingested quite a lot of the added FLA. But you can see that the red color in there is phytoplankton cells from previous meals. These are mixotrophic. They ingest phytoplankton. They take the chloroplasts out of the phytoplankton, sequester them in their membranes, and use them to photosynthesize for extra food. And these are fairly important, ubiquitous in marine systems. But we also saw some of the small flagellates ingesting the FLA. Here's a 8 by 12 micron protist, probably a very small gymnodinoid dinoflagellate, non-thecate dinoflagellate. You can see it in DAPI, it's blue color. You see this lighter colored is where the food vacuoles are. And here, switching filter sets, you can see that's ingested 6 FLA. So in our various studies, we saw other examples of dinoflagellates, not only ciliates, but dinoflagellates ingesting phytoplankton. Here in heterotrophic dinoflagellate with ingested diatoms. In the Oregon upwelling system, we saw some spindle-shaped heterotrophic dinoflagellates. We had a nice universal filter set in Oregon so that we could see both the DAPI and the chlorophyll at the same time. And these guys have had food vacuoles full of pico algae. And then in Georgia, we also saw some red-tide dinoflagellates, which are fully autotrophic, but they're also ingest. And these red-tide dinoflagellates were ingesting our added FLA. So this is another example of protist mixotrophy. When we got a chance to move to Oregon State University, we got involved in Arctic research, which is complete polar opposite from our work in the salt marsh estuaries. And the reason we got into Arctic research is another career lesson. Read widely in the literature. Find areas of research that are relatively unexploited. And we knew that there had been very little work done in the Arctic on microbial food webs in general and on phagotrophic protists in particular. So when we moved to OSU, we intentionally wanted to get into Arctic research, and it happened during the 90s when we moved there. There were some major Arctic projects, international Arctic projects, just gearing up. We were able to participate in. So that was really fortunate for us. Now, this is a diagram of Arctic food webs that was extant at the time, and there are no protists at all in there. We added this microzooplankton thing, and this is part of what we worked on in the Arctic with our colleagues Karen Ashton and Bob Campbell. And it's true, in the Arctic there are charismatic megafauna, and we saw a number of polar bears during our research up there. This was taken from the ship. We weren't down there with the bear, but they're there. The first project we had was basically only evaluating the biomass of different microbial groups. But this was a pretty cool project because it was the 1994 Arctic Ocean section in which two U.S. icebreakers went to two icebreakers, one from the U.S. and one from Canada, went to the North Pole. The first time U.S. surface ships, not submarines, but surface ships, had made it to the pole. So that was a historic thing. We were supposed to go to Alaska and back. It didn't quite work out that way. What happened was that our ship, the U.S. icebreaker, had a broken propeller, so we couldn't go back to Alaska. But it turned out there was a Russian icebreaker. We thought, oh boy, we're all up there at the pole. Boy, aren't we great explorers. It turned out there was a Russian icebreaker up at the pole at the same time, and they were doing a children's TV show on the ice. But anyway, we all got together, the Russian ship and our two icebreakers got together, and we had a big party, and the Russians served us grilled reindeer. So we ate reindeer at the North Pole during that project, and they helped us out of the ice. It was a big nuclear power icebreaker. It churned up the ice and let us out on the eastern side by Greenland and Iceland. The next project was in 97-98, and that was a project in which a Canadian icebreaker was frozen into the ice for a whole year, and we had continuous presence up there and continuous sampling for a whole year, and that was to determine what the annual cycle was. It was mainly a physical, atmospheric, optical project, but they allowed some biologists and chemical studies on there, and we were part of that. So this is our, we had a shipping container on the ice with a hole cut in it for continuous sampling, and then labs on the ship. And this was, again, mainly looking at stocks, but we also did some whole water respiration and bacterial productivity as part of that. We finally got into looking at mesozooplankton processes with Ashton and Campbell during the Shelf Basin Initiative project in 2002, 2004, and that was a number of cruises up in the Arctic, in the western Arctic Ocean. Now, this is something one can only do when you have a cruise on the Arctic Ocean, at least until as long as there's ice up there in the summer. I don't know how long that will last. So we're collaborated with looking at mesozooplankton as grazers of phytoplankton and also as food for zooplankton. And this is the zooplankton component. They took samples with a 1-meter net. Then this is examples of the different kinds of copepods. They would pick out specific copepod species. Then they'd put them in bottles, put them in an on-deck rotating incubator, incubate for 24 hours, and then look at the time zero and final to see what the copepods had eaten in terms of protost biomass and phytoplankton biomass. And we did dilution assay grazing experiments along with that, the same water masses. We had a brief interlude again. This is where that picture of the whale jaw came from. We had a project to study North Alaskan coastal systems just off the coast of Alaska in 05 and 06, and finally our last one was in the Bering Sea, 2008-2010, which we, again, replicated what we'd done with the SBI project in terms of microzooplankton grazing as food for zooplankton. That was pretty challenging because we went up there in the spring, and the Bering Sea really lived up to its reputation. There was a lot of very cold weather. We had constant problems keeping the lines to the incubators unfrozen, the gale force winds and snowstorms. It was pretty darn cold up there. Anyway, so what did we find out? Well, we found out that microzooplankton, and particularly these heterotrophic dinoflagellates, really chowed down on the diatoms, and they could eat incredibly large-sized cells. So here's a large centric diatom, and here's a heterotrophic dinoflagellate with the ingested centric diatom cell. Here's a example of a pinnate diatom chain that was common up there, and here's a protist. I'm not sure exactly what this is, but it's ingested a chunk of a pinnate diatom chain. Now, this is an example of a picture worth 1,000 words. It's really amazing how these guys, this is one of these heterotrophic dinoflagellates with no ingested prey. And this is the same sort of cell with an ingested diatom chain. And look how it expanded its cell membrane. It's incredible. It's able to do that without popping. This is two views of the same cell. This is, you can see part of the solstice here in the tip, and this is another view of it with a diatom cell inside. Just amazing. Other types of protists. We did see other types of protists in the Bering Sea feed on large diatom chains. Not in the Western Arctic, but in the Bering Sea we observed ciliates, some ciliates ingesting diatom chains, and there have been a couple of other studies in which ciliates have ingested large diatoms. We also saw parasitoid flagellates on these pinnate diatom chains. This was also observed in the Bering Sea during summer in another study, and it's been observed in European coastal waters. And finally, we found these test state amoebae specifically feeding on centric diatoms. This also has been observed in European coastal waters. So not only the dinoflagellates, but other kinds of protists are eating the diatoms, feeding on the diatoms. So here's the one data slide I have coming up. So they were also important. I should say that in terms of grazing, the dilution assays, the microzooplankton grazing rate on average was about equivalent to that of the mesozooplankton grazing rate on diatom blooms. So it was pretty significant. So there were also food resource for the microzooplankton, for the zooplankton. So here's one result from the Western Arctic Ocean SBI from the work we did with Ashton and Campbell. So in this plot, on the bottom is the prey field in terms of the proportion of the microzooplankton plus phytoplankton, the proportion of that was microzooplankton. And this is what the copepods were eating, the 4 different species in terms of the ingestion, microzooplankton proportion of the ingestion. So the 1-to-1 line is if they didn't really show any preference. Above the 1-to-1 line is preference for microzooplankton. You see mainly it was a preference for microzooplankton, particularly for these smaller Metridia and Pseudochelonis species. So what we don't know much about, and there's plenty of work left to be done on phagotropic protists, lots of work. We don't know much about taxonomy and species distribution. The work that has been done on taxonomy is mainly on small, the nanoflagellates, and there's incredible genetic diversity among these guys, just like for the bacteria and all the other groups, incredible genetic diversity. This is taken from a paper by Dan Stecker and colleagues just to show the different kinds of diversity that we have been that the geneticists have been finding. There's this mysterious group related to dinoflagellates. It's related to known parasitic dinoflagellates. There's all over the ocean. We don't know what they are doing. Are they parasites? Are they not parasites? We don't know. There's uncultured marine straminophiles that are very diverse, and we don't know what they're doing. There's a new group of phytoplankton, the phycobilophytes, and in the haptophytes, there's a stunning genetic diversity among haptophytes. We know that a lot of haptophytes are mixotrophic. They're photosynthetic, and they also eat. And I have, in the online Google album, I have data and specific papers relating to all these discoveries. How do they survive in situ? Something else we don't know. Most of the work, the methods involved in this research are very low resolution, lots of effort going to each data point, low resolution, and there are not a lot of methods. So method development is something that we need to do. There are no standard methods for assessing growth cell rates, how they detect and capture prey. We did a little bit of that just before we retired with a graduate student, reports on the biochemistry of protist chemosensory response and photoreception. Okay. Just two other things is, if you're motivated to write a book for the right public, I really encourage you to do that. I actually did that based on our time on ASLO. And in this book, it's a natural history of the Georgia coast. I put in a lot of microbial stuff and effects of climate change on the environment. But it was an incredible amount of work. So I only was able to do that on retirement. But if anybody is motivated to do that, I really encourage you. It's a great way to relate your research to the public. The other thing is, as we all know, going forward, all of our studies are going to have to take into account the effects of climate change on natural systems. And we really have to be responsible for accurate presentation of these research findings to public and also to politicians. And it's amazing to us, but we know two colleagues, skepticism about the basics of climate change among even our colleagues in aquatic ecology. And as an example, here's an exchange I had last fall with our long-standing ecology and I guess a sort of friend. So I was talking about the hottest October year in record. We've been going around climate change for a while. He said, well, Eve, I believe the temperature data. I just do not believe there's a direct correlation between CO2 concentration and temperature. Anyway, I sent him 2 good websites, and that's on the online too. And I haven't heard from him since about that. And finally, I'd just like to thank all of our people, our mentors, our students and postdocs, and all of our colleagues. So this is Karen and Bob picking copepods in the SBI project at the 0-degree incubation chamber on the Healy, very cold work. And this is Krista Longnecker, our graduate student who initiated our nomination, and we're very grateful to her for that, and also our technicians. Thanks. What?
Oh, I was just going to shake your hand. All right, thank you everyone for coming to the awards talks today, and we will have 2 more award talks tomorrow at the same time in this room. Thanks.